Okay, welcome everyone. I'm so happy to be with you today. And my goal specifically is to help you, in a sense, review the scientific method and give you some strategies that you can use maybe tomorrow. I mean, that's my own kind of personal goal. My background is in biology, and I've been teaching for Robbinsdale School's adult academic program for about 12 years. So I teach the science and a little bit of math at night too. So I think we're ready to begin. And I was kind of musing as I was waiting, thinking, this is the scientific method in action because it's new for me. And so if we run into a few snags, we just may need to think scientifically as we solve our problems. So here we go. Let's see, I'm just doing my arrow down, which isn't working. Let's see. Heather,
Sarah, if you click right on your
maybe I
PowerPoint
just
again,
have to,
click right on your PowerPoint again, and then try your arrows.
oh,
There you go.
okay. Okay, very good. Thank you. So the session objectives for today, once again, we are reviewing the scientific method and focusing in on some of that key vocabulary. We want to look at some real-world applications and some examples for how it might appear on the GED tests. We want to explore some instructional resources. I've got some dynamite resources, and also make sure that we have some ideas to scaffold, because we can't just throw the students out there and say design and experiment. So we need to do some scaffolding. And then two, I will highlight for you those scientific practices which are addressed and are a part of the new 2014 GED test. Any questions before we continue? Okay. So the science practices, and I'll just kind of talk you through these. Some of you may have already taken some practice science GED tests, as I have. I've taken a few of them, and I have seen that with the new test, the scientific method is woven throughout. So whether it's multiple choice, whether it's the short answer, where students are expected to type in, the scientific method is addressed. And another thing I've observed, and I'm happy to have other feedback as well from some of the participants here, is that there may be 3 or 4 questions for one item, one reading, for example. So they may describe an experiment, and then as a part of that, you need to draw conclusions, you need to explore the data, and so there's much more, I would say, depth to the understanding that is expected. So not only are students expected to design an experiment or an investigation, they need to be able to look at data, look at graphics, draw conclusions, cite evidence to support those conclusions, and then note the second to the last one, understand and apply scientific theories. I would say that the content is, I would almost say, secondary. If you If you have a very limited time to do science, I would say hit scientific method really hard, and some of those what I like to call the big ideas of science, those big theories, if you have time to incorporate a little, great. If not, not to worry. So I would say most important is that fundamental scientific process. And finally, as some of you may have noted from taking a practice test, there is math. And so I had one student just like last week, an older student coming in to get her GED, and she said she just wanted to try all the tests. Well, she didn't do very well on the science, and her first words out of her mouth were, boy, there's a lot of math on the science test. And so, students are expected to know those, how to figure those statistical measures, mean, median, mode, and so on, as well as manipulate equations. So we won't be talking much about that, but that is an additional science practice that's addressed. Does anyone have any feedback from the test that they want to share? Dang it. How do I get this? Thank you. Okay, if not, we will continue and jump into the scientific method. There are lots of PowerPoint presentations, lots of materials out there for you to find and use. I'm very happy with a lot of Amy Brown's materials from Teachers Pay Teachers, and her business is called Science Stuff, and she is really pretty fantastic. She includes with the PowerPoint, let's see, let's see if this works. Oops, sorry. Let's go like this. With her PowerPoint, she includes student pages, and there are blanks for students to actually do some note taking as they go through the presentation together. I think it's very well done, and so I'm a real fan of Amy Brown's materials. And this is a free download from Teachers Pay Teachers, so very easy to access. Let's go through, just so that everybody gets that little review. I think we all got it in, if not elementary school, 7th grade science. But I want to just review especially the vocabulary for the participants, and of course, take any questions as we go, but I think we can move fairly quickly through the PowerPoint. Okay, so we're talking this process
No problem.
to explain processes in the natural world. How does the natural world work? And that's the fun of science. And so there is this organized way of, kind of an orderly way of answering these questions that many of us wonder. And so scientists are proposing explanations, and it's important that they have propose something that can be tested. So it's an organized way of using evidence about the natural world. And now we get kind of to that process, that curiosity that drives science. So there's an observation, and it could be a question as well. It's very good to clarify the types of data that can be collected, both quantitative data, the numbers, for example, could be measuring length or mass or heart rate, these sorts of things, versus the qualitative sorts of data that are descriptions, involve characteristics, maybe colors, maybe tastes. These would be the qualities versus the numbers that make up quantitative data. Okay, back to the process. Hypothesis. All the students should have an idea of that hypothesis. That is, shall we say, staking out the experiment. And as I mentioned before, it has to be testable, and we could say it's a prediction of sorts. It's a possible explanation for a set of observations. Now, we talk about the steps, and they seem to go in order and be rather linear is the implication. And for your purposes of teaching the students, I would say stick to this rather linear process, because that is how it's usually taught. That's what it is expected. I will mention some ways to kind of talk about the more fluid nature of scientific work a little later in the presentation. So an observation, maybe a question, starts the process. Why does this apple turn brown after I've cut it? And what's going on there? And so then a hypothesis is formed and a possible explanation for this occurrence. And then a controlled experiment needs to be set up, data collected and recorded from which we can draw conclusions and see if our original hypothesis, our guess, our educated guess, so to speak, was correct. So let's talk a little more about the hypothesis or observation. Well, actually, step one, whether we call it an observation or a question, that is what we're starting with. So here's a few examples, and from there, then we want to form the hypothesis. Is everybody good so far? Everybody with me? I ask my students that all the time. Are you with me? And hopefully they are. But thank you very much, Catherine and others. So the hypothesis, that possible explanation. And as I mentioned before, you can think of it as a prediction. And I would just mention a few suggestions as far as writing a hypothesis. You could simply start students out with a "I predict" statement, or sometimes I suggest that students use what we call the if-then format. If I raise the temperature then the heart rate will increase. That would be an example. So sometimes having those, if you want to call it like almost a sentence frame or just a kind of a starting point, will help students with that hypothesis. But just writing it as I predict is fine. Continuing, designing the controlled experiment. And we call those factors that are involved in our experiment variables. And some of those variables are listed there for you. Could be the amount of light present, temperature, concentration of solutions. And students will be asked on the GED test to design a controlled experiment. And so what that means is that one variable is addressed at a time, and all other variables are held constant. And in that way, we can see if the variable that we changed is responsible for any changes we measure. So the controlled experiment is key. And notice that these little blanks that the students are shown, this is what they have on their worksheets. So they can, in a sense, take notes during the presentation. Okay, so we want to distinguish our groups, control group versus experimental group. The experimental group is the group receiving the treatment, exposed to the variable, and the control group is the group that does not receive the treatment, so to speak, or the change. And so that control group then serves as the comparison. Okay. So an example. In order to test the effectiveness of a new vaccine, 50 volunteers are selected and divided into two groups. One will be the control group, the other the experimental group. Notice that we want to make sure that the pill given is going to be identical in terms of size, shape, color, and texture. This is a part of the controlled experiment, making sure that these factors are held constant. And then our control group is there for our comparison, and they of course do not receive the vaccine but do take the pills. You could at this point even mention a double-blind kind of study where the patient, in this case, since we're talking about a vaccine, the patient does not know if they're getting the medication, the true vaccine, or whether it's a placebo pill, and even the researcher does not know either, so the double blind. And then the experimental group, of course, getting the vaccine. And what do we hold constant? All those characteristics of the pill. Okay. And what is being changed? Whether or not that pill contains the vaccine. So let's put names on these variables. The independent variable is the variable that is being changed. And sometimes it's helpful for students, for me to say, I for independent variable, that is what I change in the experiment. While the dependent variable, the responding variable, is the one where D for data and dependent variable, where the data is collected. So sometimes just using that first letter for either independent variable, what I change, and dependent, the data collected in the responding or dependent variable can be helpful. Okay, so in the above example, what is my independent variable? Sometimes this is called the manipulated variable. It's what I change, and it's the addition of that vaccine. And then what am I measuring? Well, I want to see, did the vaccine work? So I guess my dependent variable then would be the health of my participants. Okay, so any comments about that little example? Okay, well, let's continue. Recording, analyzing data is another big part that the students will be responsible for, not just on the science test, but the math and social studies tests as well. So data is collected and analyzed, and a part of that analysis is to determine, draw a conclusion, does the data support my original hypothesis? Okay, if it does not, I think everyone would agree that the experiment is not a failure if the hypothesis is not supported, but certainly it, whatever that conclusion, often leads to either replication of the experiment or some modification of the experiment. So replication is a key part of the experimental process. I like that on this particular presentation, Amy includes theory, because we throw that around a lot, and I use that term a lot for the big ideas of science. Cell theory, gene theory, plate tectonics theory, evolution theory, atomic theory, and so for students to realize that the meaning of theory in science is a little different from how we might use it casually in our everyday conversation, and that there is a lot of evidence to support our scientific theories. So I think we'll stop at this point, but just know that within this PowerPoint, now the students have a little practice problem where they can design an experiment from the hypothesis all the way through to a controlled experiment and draw conclusions. Any questions that anyone has at this point? Okay.
Feel free to jump in, folks, either by raising your hand and unmuting yourself or typing in the chat.
Okay, so I hope that wasn't too laborious. Let's look at a couple of ways that you can practice the vocabulary of the scientific method. First off, The Simpsons. And I have to say I'm not familiar with The Simpsons, but I found this and I thought maybe students would find these, the situations are a little corny, but they might be somewhat amused, and they're very short little scenarios and easy for students to identify the control group. What is the independent variable? What's being manipulated? What is being measured? What is my data collected? That responding or dependent variable, and what's a reasonable conclusion, and also how might the experiment be improved, which is a little critical thinking in there. So, does anyone think that their students might find these scenarios amusing? Okay, I'll take that as a maybe. Continuing, here's, totally relatable. Thank you again, Catherine. Let's go to another couple of historical cases. One has to do with figuring out the cause of beriberi. And so this goes back to 1887, and the scientists that had the good idea and tested it out. Another fascinating story is how penicillin was discovered. And I really love this story because you can get some great little videos about Alexander Fleming and how he, some of you may know the story, he dashed off in 1928 to go on holiday for a couple of weeks. And supposedly he wasn't a very tidy fellow, a very tidy scientist in the lab, so he threw a bunch of petri dishes into a sink and just left them for a couple weeks. When he came back, now if he was not a good scientist, he might have actually just given them a toss, but instead he actually, as he was kind of cleaning them out, he took a close look, and that's when he noticed that there was something, that some mold that had grown in one of these petri dishes had this ring of clear area around it. So the bacteria had not grown around that mold, and that led to his discovery of penicillin. So it's kind of a fun story, and you can find some great little videos about that. Okay. So here I just included my little tip about how to write a hypothesis and how to remember the variables. Okay, continuing. Let's see how we're doing on time. My favorite graphic organizer is also from Teachers Pay Teachers. And there are a ton of them out there. Whoops, sorry. I don't want to go to my Google Drive. Hang tight, everybody. Let's see, here we go. Okay, so the scientific method graphic organizer. There are multitudes that you can find. This is my favorite because it kind of guides the student as they're looking at it. They don't have to kind of remember, you know, what again is the hypothesis? Well, it's my prediction. And notice that under the box for experiment, it includes those important variables. What is the independent variable? And it even says, what do I change? The dependent variable, what's the data collected? What do I need to hold constant? And then there's a little box for results and then even conclusion, where you can actually evaluate, was my hypothesis supported or not? So this one's my favorite. And you may find another favorite, but you might want to start using one of these because, again, it's that idea of scaffolding and really laying out that, that experiment. Okay, so let's see, I think I need to do this. So thank you for being patient, everyone. I took this example from Kaplan, and some of you might be familiar with it. It's the electric shocks looks at the office scenario. So it comes from what's called the science practice questions. And so this is a really nice little page 506, within this series of practice questions, because they give you a scenario, actually a little everyday experience or experiment that a woman came up with, and then 4 questions to have the students consider in evaluating. Well, I thought it was perfect for using the scientific method, or the graphic organizer of the scientific method. So hang tight, and I will just rotate this one around. You're going to get tired of my graphic organizer. But basically, my students walked me through this. So why do I experience the shocks? Let's see. Just double checking here. And I'm just going to digress for a moment to show a page that I literally threw together in 5 minutes just off the internet, but just a page about static electricity and a little review of atomic structure. Here's the atom, those negative electrons being held around the nucleus of an atom, and those electrons getting rubbed off and then being discharged when we touch something. And so even some little experiments you could do, and a little bit on the history. So that was a single page that I included as a little bit of content for the students. So back to our experiment from the workplace. The manager who was asking the question, why do I experience these shocks touching the file cabinet when my coworker does not? And so the students kind of walked me through this, and we filled it out together as a part of that scaffolding. So the hypothesis we came up with, I predict that wearing, backtracking just a moment, since we didn't really do the reading, but she noted that she wore nylon clothing a lot, and her coworker wore cotton. So her prediction was that maybe it's that nylon clothing that results in the shocks. Here was their little experiment. They were going to either wear the cotton or the nylon for a good week, then do a switch, and their data, what they were recording, was the shocks at the file cabinet. So we were even able to stick in a tiny little data table here under the results, and under experiment, again, we come down, what was my independent variable? What did I change? It was the clothing. What did I measure? The dependent, the number of shocks. What did I need to hold constant? Right away, my students were talking about, well, what about the shoes they wore? So they're pretty savvy on picking up these variables. They just need this kind of organizational scheme to kind of lay it out. And then finally, the conclusion, based on the data, it looks like just a pretty significant difference in terms of shocks. Wearing nylon clothing results in more electric shocks than cotton clothing when touching the file cabinet. And so her hypothesis was supported. Okay, I think we've spent enough time on that. Is that something that you think you could use? Maybe you've already run across it in your use of or searching for materials to use with your students. Okay, I'm going to keep kind of buzzing along, creating an experimental design. This is something that, let's see, I may not have my graphic organizers for this one. I think not, but that same old one that I like so much. I had students do with something that came from New Readers Press. So New Readers Press takes you through a couple of the types of writing short answer items that they may find on the GED science test. So it could be cite evidence from the passage to support a particular conclusion, or the other one that I've mentioned, which is design an experiment. So we use the example from New Readers Press about a particular software guy who wanted to see if his online tutorial would help students improve their math scores. So we took the tiniest little prompt and we laid it out in that same graphic organizer. And the students were really good at kind of picking up, you know, those different elements of the process. Okay, let's move to, let's see if I can, okay, good, this popped up. This is from another favorite website called the Biology Corner, and look what you can give to students. You simply print these off, and these scientific or science related questions, and cut them into little strips, have students work with a buddy or in a small group, and whether they use the graphic organizer or not, it could be helpful to design an experiment. How could I test if aspirin keeps cut roses fresh longer? How could I test if hot water freezes faster than cold? And so these are kind of fun. And then once they map out their experiment, then they can share that with the group. So kind of some reinforcement, some collaboration that we try to encourage. And so let's head on back. That's something that I like from the Biology Corner, which just has a wealth of information. If you go there and just click on scientific method along the left, you'll get tons of ideas. Back to some scaffolding strategies. We've talked a bit about that and using the graphic organizer. In terms of just getting the students to write a little bit and practice their summarizing and main idea skills, I sometimes will just, oops, let's see, hang tight, will grab an article off of pbslearningmedia.org. That's another of my very favorites. So this one, for example, how cancer cells grow and divide. And I basically reformatted the essay that accompanies the video. So you've got a great little video, scientists in action, and then you've got this article that you can break it down into paragraphs and have the students basically just write a main idea statement for each of these paragraphs, and then combine each of those main idea statements into a summary. Okay, so any other comments? I know I'm kind of zooming along here, but if anyone would like to chime in, feel free. Oops. Okay. I also want to mention that, the graphic organizer we've used and used and used, of course. I also created a little page I guess it's my version of sentence frames, which I don't have a lot of experience with, but designing an experiment. So I felt that in a way this kind of guides the students through the process, and then from this they could then take those sentences and put them together to make a paragraph of how they would design an experiment. So I remind them what the independent variable and dependent variable is, what they need to hold constant. And I guess I wanted to write this out because I felt like students could get mixed up between the word control and controlled. So we say the controlled experiment, but then we talk about a control group. So we use it as a verb and a noun. And so I just wanted to kind of frame it a little more, the variables that are controlled for, that are held constant, and on and on. And then experimental, control group, the data, and the conclusion. Was the hypothesis supported or not? Okay, so a few scaffolding ideas. Continuing on, I just want to highlight a couple of great resources. One is from UC Irvine in Southern California, and what you're seeing here, if you're not sure, these are little fruit flies. A great animal model for scientific research, and I would challenge all of you to watch this video. It is very, very exciting. And I'm just going to kind of just talk you through a few things. If you just had a couple of sessions you wanted to cover scientific method, this is one thing you want to include because they walk you through the whole process. And you see student scientists in action. So this researcher from UC Irvine has been studying aging for about 20 years. And I actually went to a few summer science institutes out at UC Irvine when I was living in Southern California. So I think it's a pretty nice place. But they basically use this current fantastic research with the students doing the work and walk you through the whole scientific process. So they start with an original question. Do groups of organisms that reproduce when they're older live longer than organisms that reproduce when they're younger? And they just walk you through this initial hypothesis. Might organisms that reproduce later live longer? And so the animal model again was the fruit fly. And there it is. And why the fruit fly, you might ask? Well, it's that short generation time. So from egg to flies, 14 days. And another beautiful thing that is described here is the research piece. And here's a student checking out the website on Drosophila, the fruit fly, and finding that, oh, the entire fruit fly genome has been sequenced. And so there's just a lot of good stuff that's covered. And so again, they're using this real life research to explain the process. And it's so beautifully done. And they have all these 7 different populations that they've created over a span of a couple of years. And then they explain what they're expecting to see, these longer lifespans with the populations that are reproducing later. And the standard is that 14-day they're using as a control. They talk about replication. They have these for each population, they have multiple little chambers where they're monitoring them. And here, one of the student scientists shows all of these. There's like 7 populations with 1,000 per chamber, and then for replication, each population has another 5 chambers. That's 35,000 fruit flies that they'll be counting as they die. So it's quite fascinating. Here's the students in action. They consider, what do we need to hold constant? Well, temperature and lighting, and also food. So here the students are, boiling up a batch of fruit fly food that will be consistently given to the fruit flies. So it's just too dynamite. And again, it uses the vocabulary, it's engaging, it's got the young people doing the science, and I just love it. And so here they are counting out, those dying fruit flies. Okay. So then the research isn't done, but they even talk about the possible results. Will they see a relationship, a correlation between the age of the population and the population reproduction age with the lifespan? Or might they see where the hypothesis was not supported and the data doesn't seem to show a correlation? So really pretty dynamite. And then at the very end, the student scientist is interviewed. So it's very charming, very wonderful. Go for that. Buzzing along, no pun going there. There's another little video clip that is about 4 minutes. If you had, done a bit with scientific method and just wanted a reminder video, I would encourage you to play this one for your students. Oops, I might not be able to access this one, but it's a YouTube video, and the kind of charming thing about it, well, first off, it talks about the method, but it talks about the reality of science. It's not always a step-by-step linear fashion. And so I encourage you to watch this video as well. Just a reminder, though, for the GED, the students will need to know that more linear process, but it's good to touch on this as well. Also, the end of this video, this little 4-minute video, uses a real-life situation. My laptop, I have no internet connection with my laptop, says this young person. And so she starts hypothesizing, and then she, conducts her little experiment of, checking plugins and checking the router and the modem and so on. And it's just really kind of a cute everyday problem. And so after I'd shown it to my class, I had this older gentleman say, well, I would do those steps. I might not use those terms, meaning, he wouldn't use, well, I have a hypothesis. So it was really kind of fun. They got into that. Just another favorite resource is pbslearningmedia.org. Love it, love it, love it. This is an entire lesson. And if you don't already have an account with PBS Learning Media, you must get one and then create a little file of your favorites. This is too great. It walks you through and has at the ready all these different little videos about the scientific process. And it also kind of gives you the reality of the fluid nature of science. And so students in little groups, like of 2 or 3 with a laptop or a computer, they could watch a particular video. That's the one their group is responsible for. And then the next group has another video. And basically they'll, come up with any of the science processes, science elements that they saw within that video and share it with the group. So it could really be great. Now, of these videos, I think I've watched Stem Cell Breakthrough, Synthesizing an Alkaloid. Let me just say this one is dynamite. And it's this Percy Julian, who is an African-American scientist. I can only imagine what he was going through in the 1930s as a Black scientist. And it's about the goal, which was to synthesize a drug that could be used to treat glaucoma. And this other guy had, it was kind of a race, which often it is in science, but this other guy had come up with this long, drawn-out, million steps to synthesize this compound. And Julian Percy, or maybe it's Percy Julian, sorry, he comes up with a more simplified version, just minimal steps. And it all came down to the melting point of this material. And he found when he tested the melting point on this other guy who had just published his scheme that this other guy was wrong because it wasn't the actual compound. And so Julian became the first guy that was successful doing this, and his process was much simpler and more elegant. So just a kind of a kudos to that video. But I wanted to show you at PBS Learning Media how you just click on the support materials. You've got a background essay. This is what I reformat sometimes for the students. They even include some discussion questions. So PBS Learning Media is the best. Okay, back to, this was another one from PBS Learning Media. Blood vessels help tumors grow. And just fascinating about how, this doctor really believed that blood vessel growth had something to do with why the cancerous tumors grow so fast. But other scientists and doctors didn't really believe him, and he had to find the mechanism. And it's just quite a fascinating little story. Okay, so moving along. Nobody's interrupting, so Sarah is off to the races here. Another important skill, of course, is graphing and analyzing data. So I've got a couple of nice little labs you can use. One is, oops, I guess I may not be able to open this one, but not to worry, it will be on the resource page. And this is about a neurotransmitter in the brain, and this idea that if you have more of this dopamine around, you may want to take more risky behavior. So they're making the link there. And it's about a research study, and they give you a lot of data, and the pages to print out are beautifully done. They give you the nice little graph paper section there so you can graph the data and draw conclusions. I love it because they're plotting those points, they're making a scatter plot, they're drawing a line of best fit and making conclusions from that. looking at the overall trend in the data. So the other one that I like very much, and this is Amy Brown again from Teachers Pay Teachers, and this one is a free download. Let's see if this works. Let's see. I think I'll just, we basically have a little picture of it, but again, free download, and with this, it's a fun little activity. If you have a scale, if you can weigh your pieces of cardboard, basically what you're doing is for each group, you cut some different rectangles of this corrugated box, and then from there you also want to measure the mass. So if you have a scale, you can weigh them as well as measure length and width and use the area formula. And so then you can plot on a graph the mass to area, and it's very nicely laid out. Draw your line of best fit. Then you give the students an irregularly shaped piece of that cardboard. And for me, it was kind of amusing. One of them I had made, I didn't intentionally do it, but it was kind of the shape of the wine glass. So it was kind of funny that the one group, was always referring to their wine glass. But so then from that irregular shape, they find the mass of it, they put it on the scale, find the mass, and then they can read their graph, read up to that line that they created on their graph, and then from that read back over to the area. And so that is some good practice too. Okay, I'm not going to say too much about heavy-duty content, but one of my new ideas this year, because I have this favorite evolution book, and okay, maybe I just guess I'm not, oh yeah, I'm going to take it from my Google Docs, if you don't mind. I decided, because this is my very favorite evolution book, and it explains natural selection so beautifully by using frogs, and the artwork is so glorious that I thought, well, maybe I can do something with these picture books. And so I actually found several of them, a whole handful of these books at Hennepin County Library, and brought them in, and then had the students just kind of evaluate and see what they liked. And here I had to include my favorite pages where it actually explains natural selection so beautifully. So you start with the 3,000 eggs from the frog and go through the tadpoles, and how ultimately there's only 10 little frogs that make it. And out of those 10 frogs, it's the good jumper and the one with good eyesight that survive. So it's got it all. And I love the art, so that's kind of fun. Maybe that is something you'd want to try too. Then I've also have just identified here the best PBS evolution series ever, and you wouldn't have to necessarily go through the 7 videos, but they are just excellent. And I would for sure hit the first one. Isn't evolution just a theory? So they really explain why theory in science is different, how it is different. Then another video that is quite good is in the series is how does evolution really work? And they go to Ecuador with the scientists who are studying the hummingbirds, and they're making the measurements. They're drawing the blood for genomic studies. I mean, it's really science in action, and it's fantastic. And they explain it so beautifully with these hummingbirds and bill length and flower size and all of that stuff. The other video that I love very much is this number 5 in the series, where they go into a prison over in Russia. Scary place, but they basically are looking at how TB has evolved and become drug resistant, and how this population is actually a pretty good population to study this. So, and then the final one is, why is evolution controversial anyway? And that's a really good one too, because I have some of my students who can be they don't really want to learn necessarily about evolution, but it is central to biology, and so I do give them a little bit of that. Okay. Any questions? Anyone want to interject before I just highlight a couple of Dynamite resources and websites for you? Heather, could you say hello?
Hi, we can hear you.
Okay, I suddenly had this moment of panic. What if I've been talking to myself all this time?
You have
So,
not. We are still here.
Okay, great. Let me just scroll down. Is that okay with you, Heather, if we just highlight a few of these Dynamite websites?
Absolutely. We've got time to do that.
Okay, super. First off, oops, let's see. Maybe I need to do it, let's see, this way. Yeah. Okay, so this Utah site, Learn Genetics, is too amazing. I mean, if you really wanted to just let students explore on their own and just give them a topic, you could. In terms of genetics, it's got all these fantastic little, what they call tours, where they guide you through what is heredity, what's a gene, what's a protein, and so on. Sometimes they call them interactives, where you go right inside a cell here and just kind of roll over and look at some of that interior structure of a cell. Oops, thank you. And so anyway, this is a really pretty fantastic website, and it's got a lot of the things that students are hearing about: stem cells, cloning, and so on. A little bit on genetic disorders, and some virtual labs. So I would say even some mathematics for Rebecca Strom. Okay. And you can see that I'm a little clumsy here, but everybody seems to be hanging tight. Another very great, cool interactive, if you want to just touch a little bit on some of these big ideas, from PBS Learning Media, here's a little tutorial on atomic structure. And I even put this on the big screen and on the smart board, and just, reviewed atomic structure this way. Because you start with that tip of the pencil, and you see that those vibrating particles, and you zoom in and it appears fuzzy, that little atom, and then you see some of those subatomic particles, and so on, and it goes through those different parts. So really great. Another one of my favorites is TV 411. So I don't know if anyone's used this. I've really just used it for the science, but I am guessing that the other areas are good too. Um, this can be kind of fun. It can actually be used, both the science and math lessons, um, with or without the accompanying video. Because, for example, let's take this one on heat, which gives you a little bit of content in the energy and the electromagnetic spectrum. Well, you always get a recipe, and this, cooking in the kitchen to start out. And usually the students like that. But it's not just the cooking part. They actually do some science in these videos. And then you click on the science lesson, and it walks you through. So I've had students use a laptop in partners, and it's just really great because they teach by giving you just a little bit of text and graphics, and then they ask you questions, and you go through with your partner, and if you get it wrong, then it'll lead you to the correct answer. And then, there's usually several activities in here. And here they're talking about the electromagnetic spectrum. They're doing some drag and drop as they do on the GED test. And then finally, after you do several questions there, then they will quiz you with, about 10 multiple choice. So the students tend to really like this, and I think it's great. And this way you can, get a little bit of your chemistry and physics in there. Some of those ideas. Okay. Plate tectonics, another big idea in science. Here's an entire lesson from PBS Learning Media. And notice they have all these little videos selected for you with the lesson plan. So, you can kind of pick and choose, but I think it's nicely laid out for you. Oops, I think I should be using my Alt tab, sorry. And now a few that I have just been kind of researching of late. Climate change, if you go to the NASA site, you've got some unbelievable graphics. So just having them doing some graph reading is really pretty great. The other one is this student guide from the EPA site. Got a great little video on climate change basics. What is the greenhouse effect? And see the impacts. And I actually wrote a little worksheet to go with this. And I was a little sheepish when I told the students, that it was the student's guide to the global climate change. But it's just so nicely laid out. And the information is so good that I think they enjoyed it too. And then there's some little questions as they go through. So excellent resource. Okay, so I have to include one more little thing. And this I just did recently with my students. So this came from Teachers Pay Teachers. And let's see, oops, sorry. Okay, I think I don't, let's see. I just want to show you real quick, if I can, sorry. Okay, so this was a little worksheet that I made, and I didn't really plan to do this on this little microscopic creature, but I decided that, and here's the Daphnia. You could find it in some pond water that you scoop up, and it's used a lot in toxicology research. So it's almost, well, it's used as an indicator species. It's very sensitive to different chemicals. And so when I saw some kind of a graphing lesson from Amy Brown, and I did have to pay for this one. I don't know as I can kind of get that to you, but it's as I show you here, it's a series of 3 different great graphing exercises, and it reviews the scientific method. It's fantastic. You'll have to pay $3 from Teachers Pay Teachers, and I was glad I did. So example 2 out of the 3 is the one on the Daphnia heartbeat. So the students can graph some results and then go on to design their own experiment. And so I just decided, well, let's introduce them to Daphnia first. And so I had this, I gave the students this little sheet that I kind of threw together, and I had them watch the video and then stop it and then sketch. And the artwork, the detail was astounding. And then I had them, they were using iPads, then I had them find an image that where they could label the various parts, find an image off the internet with the parts labeled, and then record some observations, and then even count the heartbeat, because they get this little not even 2-minute video going, and the heartbeat is really fast, but they're working with their partner, and they really had fun with this. And then write 3 questions. So you've got this where you introduce the Daphnia. They kind of get close to the organism, like it a lot, and then they do the graphing part and draw some conclusions. The final thing, which I had different student tables do. One table was the nicotine group, another table was the alcohol group, and the other table was the caffeine group. And their challenge then was to design an experiment to test the effect of those chemicals on heart rate in the Daphnia. So they'd have to write their hypothesis, what's the independent variable, what's the dependent variable, what should they hold constant, and you know, just kind of lay out an experiment. So that was a lot of fun. The biology corner, I referenced this. I would encourage you to go there. There's all kinds of, you know, simple labs, and also a lot of graphing practice. So it's a really good site, and you can kind of see at this opening page on the left-hand side, scientific method, that you can click on that. Okay, this is a Guide to Understanding Science 101 is from the Berkeley site. And this too gives you a different flow chart in terms of the scientific method. Not so linear. Lots of, you know, back and forth and retesting and so on. And it kind of dispels a lot of the misconceptions about science, and it's got some really great things on this Berkeley site. Okay. So as I look down, I think I've mentioned everything except for scientists at the Smithsonian. And this could really be fun. I haven't used it a lot, but you are with a scientist in the field. Here's a dinosaur hunter. Here's a bee tracker. Here's a monkey monitor. And this is really fun to see these scientists in action. And they're doing the work and following the process and answering those questions about the natural world. So very exciting. And now I think we have a little time for some processing, reflection, or questions. Would anyone like to share?
You can chat in the chat box or raise your hand. We can unmute you. I'd love to hear what people are thinking. If Sarah's presentation has sparked any new ideas, or you have other resources that you have used that others might benefit from, please do share. Or ways that you might adapt what you heard about to your own context or your learners.
I'm curious to see if any of you like the graphic organizer. Could you see yourself using that?
Seeing some yeses coming in on the chat box.
Okay.